Today we're going to be talking about macromolecules, and this presentation will be broken up and into two presentations. We will focus on key characteristics of each of the macromolecules, and you can start by studying and filling out your chart and your notes packet as applicable. So as every lecture, we have learning outcomes. In these learning outcomes, this will be a way to start to guide your studying. However, this is not going to be all of your studying. So use this as your guide to study the notes. But we will first start by talking about carbon. And carbon is the framework of a lot of biological molecules. And specifically, we're going to see this list of functional groups and structural formulas right here, and the example and how they're found. So you can see how we have hydroxyl, carbonyl, carboxyl, amino, sulfhydryl, phosphate, and methyl. And what we're going to see is that we can form up to 4 covalent bonds with this carbon molecule. And if you remember from our chemistry lecture, you'll remember that covalent bonds are strong. Double covalent bonds are stronger. And this is going to be really important when we start talking about biological molecules and its applications. This is really interesting to me because I think when we start talking about macromolecules, the application of biology goes much farther, or it can extend past a lot of content areas. And so when we talk about the macromolecules, it has very great implications for a lot of topics in biology. And so when we have a very good, healthy understanding of macromolecules, it can really bridge a lot of the gaps. So you can read in other content areas a lot of content that you would have never been able to synthesize or understand or acknowledge before, which I think is really good as a critical thinker. And so when we talk about carbon and how they can form these carbon or those covalent bonds, we then can start talking about drug deliveries and hormones and all of these different things that we never could before. And so when we look at this, we also have hydrocarbons, and this is where we have a molecule consisting of only carbon and hydrogen. This is a nonpolar, and they will have functional group chemical properties. Here's a picture showing what this can look like. We have methane, ethane, and ethene. And you can see how the different bonds with carbon can yield the different shape of different molecules. Different shapes mean different molecules as well. And it goes back to understanding valence electrons. So when we have hydrogen, oxygen, nitrogen, and carbon, we can quickly see that the outer shell in carbon has only 4, so 4 on the dot diagram. So this is going to be really helpful to form the skeletons of these organic molecules. And these carbon chains are going to vary in length and shape. And when we see this with varying in length and shape, they are different compounds, again, like I said. And so we have ethane versus propane, butene, 1-butene versus 2-butene. And the 1-butene is because the double bond is on the first carbon. The 2-butene is because there's a double bond coming off the second carbon. So all of that information we learn in organic chemistry, but we can start applying our knowledge now here. And then we have butane, and then isobutane, cyclohexane, where we can see it's a ring, and we will talk about rings versus chains. And then benzene, and we can see how there's double bonds. There's 6 carbons, and then there's double bonds or single bonds, cyclohexane versus benzene. So then we need to talk about functional groups, and these are going to be really important as they relate to these organic molecules, and they will depend on its number and arrangement. So this is what's going to give these chemical compounds specific properties. And we saw this in the last slide here, how we have different properties or different names associated with these chemicals. An example that we would see of this is estradiol versus testosterone. You can see how we have a hydroxyl coming off this carbon molecule, not labeled with a C, but we know it's a carbon compound, versus this double bond to O right here. And we can see there's also a methane with testosterone. Well, interestingly enough, estradiol is in birth control and female replacement or hormone replacement therapy for females. And so we will see that estradiol can actually not be filtered through our wastewater treatment plant. Well, if it's not filtered through their wastewater treatment plant, it's got to go somewhere. So where does it go? It goes into our wetlands. And what we see is we have these estrogen dominant systems then quickly happen where we have frogs that are immersed on land and water. They're immersed in water, but they can live on land, and they are exposed to this estrogen heavy water. Well, when they are exposed, just like women are taking birth control to regulate their menstrual cycle, we can see that we have this induced female frog that is actually a genetic male. And so it can actually change the population chemistry of the male, and so the chance of them mating or having males in this system right here that's estrogen heavy is less likely. So there's a lot of information starting to come out about how we have estrogen hermaphrodites in the suburban waste areas. And in the PowerPoint, I also linked some articles. So it's really interesting to consider. And this is where we start quickly getting into these context areas of what happens when there's an oil spill, what happens when we're not filtering everything from wastewater treatment plants, and what does that even all look like? So it becomes a lot of fun to kind of research and start talking about some of these topics quickly. Another thing we can see here is we have 7 functional groups. There's a hydroxyl, carbonyl, carboxyl, amino group, sulfhydryl, phosphate, and methyl group. We'll talk about each of them. So the hydroxyl group, we can see that we added this. The functional properties is it changes the electronegativity, and it can form hydrogen bonds with water molecules. So this is going to be really helpful to dissolve. Carbonyl, we see that we have this C double bonded to an O. And an example of this would be a ketone and an aldehyde. So we see this with acetone. Acetone is what's found in fingernail polish. Carboxyl, this can act as an acid because it can donate this proton. And so we see this be carboxylic acids and organic acids. And so, yep. Then we have amines, and this can act like a base. So going back to pH, it can act like a base and can pick up these protons. And this is going to be really glycine. This is going to be really helpful for cell walls. Sulfhydryl, we talk about this this is a big one that we talk about with our body, our skin. Sulfhydryl, you can see as this SH. This is going to be this is a cross-linking that will help stabilize protein structure. So you can kind of star sulfhydryl in your notes that we will come back to this. This is also going to be one of those proteins, or it will help with a protein hair proteins that will deal with curly or straight hair. So we can reform these bonds and reshape these bonds by perming or straightening our hair depending on the hair curliness or straightness that you have. Oh, phosphate, organic phosphates, PO4. So we have PO and then there are 4 O's. So we say this as PO4. It is 2 negative, and we see the 2 negatives by the 2 oxygens at the top. And then this is going to be really important when we start talking about this ability to react with water. It's going to release energy. It's also found as part of our membranes. It's also found in our DNA. And so this is going to be a really important molecule as well. And then methyl, this is a CH3. You can see this. And it's going to be bound to DNA. It's going to be found in the expression of genes. And then methyl groups, we've talked about this already. We saw that with estradiol versus testosterone, so that's going to be important to consider as well. So before we go farther, we do need to talk about ATP. ATP is adenosine triphosphate. It has 3 phosphate molecules here, and what we see is that we have energy transferring molecules. We have adenosine, which is the main molecule, and then 3 phosphates attached to that. And what we will see is when we break off a phosphate, we will have ADP, adenosine diphosphate. And then we will be able to, when we break off a phosphate, we actually release energy. So when we go from ATP to ADP, we're going to say that we're going to release energy. So it's like breaking a glow stick. So how do we make ATP? We do this in the form of exergonic cellular respiration. We build energy back into the system to make ATP. So we see here how ATP can react with water, and then it yields an inorganic phosphate, so it drops off a phosphate, and then it has adenosine diphosphate, di means two, and then releases energy. So it's going to be really important to consider as we're going through this process of understanding these organic compounds. When we look at these four classes of biological molecules, there are four main types. I always go through it in this sequence: the carbohydrates, lipids, proteins, and nucleic acids. And we have very large molecules. And what we're going to learn, and what the key phrase here is, that shape defines function. So on your chart that we're going to use to study, there's going to be a lot of information to put in. What I want you to do is focus on the notes right now. And so here we have a chart. We're going to use a variety of charts that you can go back to later. And so slide 24, you're going to see that we have different carbohydrates, nucleic acids, proteins, and lipids. And we have a lot of lipids shown in this table. And here is understanding the cellular structure, polymer, and monomer. And first we need to look at the dehydration synthesis. So with a dehydration synthesis, we're going to actually join two monomers are joined to form a polymer. And so with a dehydration synthesis, we're going to take a H, combine it with a OH to make water, and that is what's going to allow us to actually make a polymer. A polymer is, poly means many, so mono means one, so we have many units versus one unit. Hydrolysis is when we break a polymer by adding water, and polymers are broken down into monomers. So we can see that with this example. I like this slide a little bit better. You can see a short or an unlinked monomer, and dehydration is going to remove the water molecule to form a new bond. And you can see with the carbon compounds here, 1, 2, 3, we're going to then have a longer polymer here. And this is the same here. Hydrolysis, we're going to break down a polymer. Hydro, water, breaking, lysis is what that means. And we're going to see that water being broken down. So it's going to be really important as we look at these different compounds as well. So we have carbohydrates, and these molecules are with a 1 to 2 to 1 ratio of carbon, hydrogen, and oxygen. So we have a formula that looks like CH2O O with a subscript N. And this N right here is saying that if we had an N, and we're going to use an algebraic expression here, that we're going to see that if I say 6 right here, if the N is 6, you can write this in your notes. If the N is 6, then it's going to be C6H12O6. So CH covalent bonds will hold a lot of the energy. Carbohydrates are good energy storage, and we are actually going to see that some examples of these are sugar, starch, and glucose. So monosaccharides is the simplest carbohydrate here, and we can see that it's a 6-carbon compound. We can see that it's in the ring structure or in the the ring or the straight structure, the chain. Straight structure, it's a chain. And like I just talked about, glucose C6H12O6, you do need to know that formula, is the most common monosaccharide. Fructose is an isomer, and what we say is an isomer is we move the bonds around to change so it's somewhat similar. That would be the easiest way. Galactose is going to be a stereoisomer, and so we say that a stereoisomer is something going to be where one aspect of it is going to be different. The reason why I bring this up is when we start talking about just in these three cases, enzymes in our body, and we'll talk about what enzymes are in a little bit, enzymes act on different sugars. So when we are eating sugars from an apple, right, we can say that there's fructose in apples. That is going to be a different sugar than what's in Mountain Dew. And our body is going to react because it's going to have different enzymes because of the different sugars we put in our body. And so it's going to be really important. And sugar is one of those compounds that there's a great book I have talking about the impact of sugars. And it's just really interesting of how much sugar has changed the landscape of our food and even food availability in our nation and other nations. And it goes back to this simple aspect of macromolecules and enzymes. So then disaccharides here, we see that disaccharides are linked together by two monosaccharides, and two monosaccharides make a disaccharide using a dehydration synthesis. And one of the things that you will note along the way is we will have different linkages. When we have a linkage, it's specific to this macromolecule, and it's used for sugar transport or energy storage. And so an example of this would be sucrose, lactose, or maltose. So we have sucrose and we have maltose down here and fructose, right? Glucose and fructose will yield a sucrose. And here we have some more examples. Maltose and sucrose. Two glucose make a maltose, a glucose and a fructose make a sucrose. So then a polysaccharide are going to be these long chains of monosaccharides, and they can be used as energy storage. So plants will use this as starch, and the simplest form of starch is called amylose. And so here we see that we have amylose, amylopectin, and then we have glycogen. Okay, so very more complex. And then we have glucose polymers. We have amylose, cellulose, glycogen, right here. And animals are going to be using glycogen. We have glycogen, and this is how we're going to store it in the liver or muscle cells, is something called glycogen. So here's amylose and here's glycogen. Now again, we have these plant and animal polysaccharides. So when we start talking about different polysaccharides, they do differ based on plants and animals. So when we talk about how to break them down in our body, that will depend on the enzymes. So again, looking at this, plants use cellulose as a structural support, and the glycosidic linkages will differ. Cellulose in humans will actually be as the example of insoluble fiber. So here we see starch, and we see a 1-to-4 linkage. In 1-4, we know that means something about these carbons. Cellulose is 1-4 on, this is the alpha, that it's starch is 1-4 linkage of an alpha glucose. Cellulose is 1-4 linkage of a beta glucose. So they are different compounds. And so it's just really interesting because when we look at the alpha glucose versus the beta glucose in a chain, it goes back to where are these functional groups. So when we look at the cell wall, we have cellulose, and then we have these cellulose microfibers, then we have this beta glucose monomer. And then we have arthropods that have this exoskeleton, and fungal walls, cell walls that use chitin. And chitin is going to help form the exoskeleton, so you can see how that's what they shed. And chitin is also used for surgical thread. So a great chart looking at this and comparing this, we see that we have simple and complex carbohydrates. Monosaccharides and disaccharides are going to be on that list, and then we have polysaccharides right on the other part of the list. The reason why this PowerPoint is so long is we actually have a lot of pictures in this particular PowerPoint. And so we have a polysaccharide, chemical structures, and three-dimensional structures. Our next topic that we're going to look at are lipids. And lipids are going to be loosely defined with the one main chemical characteristic that they are insoluble in water. The high proportion of nonpolar covalent CH bonds cause these molecules to be hydrophobic. You can underline that in your notes. And some examples of this, and we'll talk about a lot of these examples. So we have triglycerides, that is composed of 1 glycerol and 3 fatty acids. And glycerol is a 3-carbon compound, 3-carbon alcohol with a hydroxyl group, and we will look at what this looks like in the next slide. A fatty acid of a carboxyl group is attached to a long carbon chain, and 3 fatty acids are joined to this glycerol with an ester linkage. So here we have this polar head group. We have a choline and a phosphate, but we were talking about this glycerol, 3-carbon glycerol backbone, ester linkage right here, and fatty acids. And this has 2 fatty acid chains. This a triglycerol, it shows you this ester linkage right here, where we have 3 ester linkages, 3 fatty acids to get a tri, right? So these fatty acids, they don't need to be identical, and the length, chain, number, and locations of the double bond will vary. And this is going to be really important for us to understand, because here we have on the left, we will start to see the difference between a saturated versus an unsaturated. A saturated, you can see how we have this where they don't have any double bonds, and they are going to be, the carbon chains are going to be laid very straight and flat on top of each other. The unsaturated, if you look in the right column, you will see that the unsaturated triglyceride will have different double bonds in different places, and that will give different quote-unquote kinks in the molecule, where the fatty acid tails will not lay very close to each other. Here's an example of what that looks like, and it causes the chain to bend. So this carbon compound will actually be quote-unquote bent. So saturated, no double bonds between the carbon atoms. They have a higher melting point, often animal origin and solid at room temperature. Lard is an example of that. Unsaturated would be low melting point, plant origin, which we can't even say that anymore because we have so many modifications in our food aisles now, and liquid at room temperature. So olive and avocado oil is an example of that. Trans fats, which we have seen integrate so much into our grocery stores, is what we see where we have this quote-unquote spreadable butter. So back in the day, it was very common that a diet rich in saturated fats actually led to cardiovascular disease. However, we know that there are very strong benefits of eating things like real butter, real milk, right? Not other products that have been changed by the industry. And so I say that because it's more accepted that we actually go back to eating real butter and not using vegetable oils and corn oils. And many times I have had many students actually be allergic to a lot of these products over time, and such an increase also over time. And this is not necessarily celiacs, that is dealing with the genetic information in the wheat and the food. We're specifically now talking about oils that have been food in general that has been changed by the industry. So we do have certain unsaturated fatty acids that are not synthesized or not made in the human body. So we do know that we must have some supplied in our diet, and it must be included omega-3 fatty acids to help protect against cardiovascular disease. Now, there is a lot of information of how cardiovascular disease is a very prominent disease in the US, and that we need to be aware of it to take measures to prevent this. And so this is not something like, oh, just change your butter to just, real butter. It's a very distinct conversation that some families have to make, very distinct conversation that some families have to have very early on in their lives. So it's just something to definitely talk about and be aware of, and also see how the food industry changes our ideas about food overall versus where do we need to get the most nutritious food from that is healthy for our each individual bodies, and that is an individual decision. And so our next thing to talk about are phospholipids. So phospholipids contain a glycerol, 2 fatty acid nonpolar tails, and a phosphate polar head. So oftentimes you need to know this part. We saw this earlier in the presentation. And this is what's going to form biological membranes. And what we often draw is the circle with 2 tails. So I like the next picture a little bit better because you can kind of see how you have this phosphate and this glycerol. And we have a hydrophilic head and a hydrophobic tail. So this is a symbol, and I often draw this on the board where we have a head with 2 tails, okay? And oftentimes, mycelials are lipid molecules that have the head towards the water and the nonpolar hydrophobic tails away from the water. And these are like the fat droplets in our soapy water that didn't get destroyed. So a couple things about that then. So we also have a phospholipid bilayer. You need to know that term. It is more complicated. It looks like 2 pieces of sandwich bread maybe, and we have these hydrophilic heads. You need to know that the heads are hydrophilic and the hydrophobic tails are inward. So the heads are outward, tails inward. And we see that these fats will separate from the water because the water can actually form bonds with each other and exclude the fats. And so this is going to be a really important conversation in our next couple lectures. This key concept, this particular slide, will help do some of the framework of our understanding as we go into cells and what makes up the cells. So then steroids is another example of a fat where we have 4 fused rings. Cholesterol is found in animal cell membranes. We're animals, so we have cholesterol found in our cell membranes, and it is essential in animals. So we have high levels in blood that can actually contribute to cardiovascular disease. And so we have cholesterol, 4 fused rings. And so then we have the dehydration, so we see that this can occur. We have a glycerol with a fatty acid tail. And we understand that fats, with a function of fats in general, there is energy storage. And how humans do this is that they store fat in adipose cells. Now, adipose cells, there are 2 different types of adipocytes, and what we call as adipose tissue. So some of the adipose tissue is around our organs, and then some is actually around it's found on the outer part of our body, but subcutaneous, so under the skin layer. So, and they serve different purposes. So when we talk about this, it's really important that we're not talking about any healthy eating, although healthy eating is a very important conversation, or any fad diets or exercise. We are talking about in general as it relates to the body as a whole. What does the fats in general mean for mammals or animals or our own bodies? That concludes this is the first part that concludes the macromolecule lecture.